So I guess there's nobody introducing me, so I just give you a self-introduction. My name is Guanming Wu. I'm working in this department for doing computational biology and bioinformatics data analysis. And also, we are building this software called Reactome, which is a biological pathway knowledge base. So the title here today actually is quite big. So originally, I thought I can give you guys a general introduction about using Reactome for systems biology data analysis. But it seems like it's just too much. So today I'm going to focus on two topics. The first is just a gentle introduction about this project. Second is about its one specific sub-project we are working on called Reactome Functional Intelligence Network. So the project we have been working on for almost 20 years. So there are a lot of things I actually can talk about maybe for several days or sometimes several weeks. So in this 30 minutes, I'm going to try to focus on really give you some introduction level talk. So, okay. So before I introduce the Reactome pathway database, or we call the pathway knowledge base, I'd like to talk about why we need a pathway-based data analysis and pathway database. So why do we need a pathway data analysis? So here is a motivation example. So about 6 or 7 years ago, several research groups in the world performed a so-called pan-cancer data analysis and found this 127 cancer genes. Their mutations actually people already know can cause tumor development. So here is a list of these 127 cancer drugs, cancer genes. So from here, what you can see is what you can see. You basically cannot see nothing. So we really don't know what these genes, we know what these genes look like, of course, we know their names, but we really don't know what their function is, why their mutation can actually cause tumor development or causing cancer. So how can we really understand their functions and why their mutation can cause cancer? So here, biological pathways come to rescue. So pathway-based data analysis basically project a list of genes like 127 cancer drive genes or to pathway context. Then give us a context to link all these genes together to see what all these genes function. So basically, the first one is it can help us try to visualize genes seemingly unrelated in pathway context. So these genes can come from so-called differential gene expression data analysis. If you guys come from computational biology world, so you have done, I believe you should have done a lot of this kind of data analysis, or to come from these so-called gene wide studies for genome-wide association studies. So from gene-wise, we can get a list of so-called significant SNPs. They can be mapped to genes and get a list of significant genes. And also, they can come from epigenetic analysis. Okay, so as I said, by projecting all these seemingly unrelated genes onto pathways, basically we try to understand the mechanism why all these genes have. So they most likely, some of the genes may share similar functions. And by doing pathway data analysis, we can also try to increase the analysis power by reducing the data, the actual data complexity from like several thousand genes. Originally, you may have a lot of significant genes. Then you can actually map it to several hundred genes. So you have some kind of so-called dimension reduction. And also provide functional context, so which give us so-called explanatory power to understand all these biological implications. Yeah, you can ask questions whenever you have. So, okay, we should have enough time because originally it is for 30 minutes. Right now I have the whole 60 minutes for me. Okay, so several years ago, a group in, I believe in UCLA somewhere, so the first author is, Catcher. They generalized all pathway-based data analysis approach into this so-called 3-generation in a review paper published in PLOS Computational Biology in 2012. So the first generation is so-called over-representation analysis for ORA. Basically, was developed for doing differential gene expression data analysis. So from differential gene expression data analysis, you can get a list of significant genes by so-called hard threshold. So you can get this. Then you do some kind of very simple statistical analysis and try to figure out what pathways are significantly enriched or significantly downregulated or upregulated in this set of genes. So this is the first generation of pathway enrichment called ORA. So as you can see here, this approach, they need to use hard threshold to pick up a list of significant genes. So there are a lot of information differently is lost. So people come to this second generation pathway analysis called functional class scoring, or FCS. So in this generation of analysis, they consider every genes statistics, and then they develop some kind of statistical test. The most popular one in this generation is so-called GSEA, for gene set enrichment analysis, which is really probably the most popular pathway enrichment analysis, even at the present. So from here, but still there's something, as you can see, if you know some pathways, so pathway actually there are a lot of topological structures. Something like some genes activate downstream genes, or some genes inhibit or activate all this kind of relationship. So the first generation approach, they basically don't consider all this very rich information. So people come with this so-called pathway topological based data analysis. So try to consider this kind of topological structure, including number of reactions, position of genes or proteins, and what type of reactions involved. So one of not that popular right now actually, one of approach developed for this pathway topological-based approach is called SPIA, for signaling perturbation analysis. Okay, from here, so all these three generations, they are all based on this so-called pathway database. So from here, you can see a pathway database basically is the foundation for doing all kinds of pathway data analysis. So it's an indispensable resources for this. So come back to Reactome. So currently, there are multiple pathway databases available. The three most popular pathway databases, of course, we are talking about Reactome. So Reactome probably is the most popular one. It is open source, open access. There's no any constraint to use Reactome. So then the second, this actually probably is the most popular one for experimental biologists to do pathway data analysis, called KEGG. But unfortunately, KEGG originally was open source. You can have you could have unlimited access to that, but because of some kind of funding issues, about 7 or 8 years ago, so they need paid license in order to batch download their content from their FTP side. But as a user, you can still access the content on several web-based services from KEGG by using their website or RESTful API. So the last one I'd like to point out is called WikiPathway. So the WikiPathway, they use this wiki-style online editing tool. So basically, you can edit all pathways provided by WikiPathway online directly. So they use all this wiki-style try to tracking all this editing history. So it is quite useful. But the problem is, since the goal here for them is try to develop this as a community resources, But unfortunately, biologists seems not that willing to do all this community service because I guess they are just too busy. Okay. So come back to Reactome. As I said, Reactome is open source, open access pathway database. All content are expert authored, manually curated, and peer reviewed. So the bottleneck, just as I said, is we are very, very difficult to recruit external biologists to do some curation for us in a timely way and high quality way. So, but since we are doing our best, trying to really collect data from all these experts. Okay, and all pathways in Reactome are actually supported by original experimental evidence. They can be traced to the preliminary literature. Currently, we have annotated close to 30,000 published literature references. So that is quite a lot. So the data in Reactome is extensively cross-referenced to external bioinformatics or chemoinformatics database. So as a community resource, this is very important. So people can cross-reference together and see what actually happened. So imagine in the future if there's some kind of really artificial intelligence or machine learning power system, so they can cross all together to generate a unified knowledge system for users. So we also do some so-called computational inference, try to infer pathways from human to other non-human model organisms. Of the manual focus, the manual curation, the focus of our manual curation is on human pathways. So we collect all this experimental evidence from experimental systems for human biology. So, but in order to reach to a wider research community, we do this so-called computational inference. And also, along with the data, we also provide a suite of user-intuitive, high-performance software tools for users to browse our content and perform some pathway-based data analysis. So I'm going to show you more about this. Questions? Okay. So Reactome basically is a so-called ration network database. So the foundation of all annotation is biochemical ration. So the biochemical ration is the basic unit in our data model. So this ration-based data model actually is very flexible and versatile. It can be used to annotate almost all biological processes currently people have found from molecular levels inside a cell or between cells. So this is great. And according to biochemistry, we annotate a ration as a set of inputs converted into a set of outputs. And this conversion can be regulated by regulation. So this regulation includes activation or inhibition. And of course, this is a biochemical reaction. It should be annotated by some kind of catalyst. So we have this so-called catalyst activity. So as you can see here, all objects annotated for a reaction and also reaction itself is extensively cross-linked to our external database, as I just pointed out several slides ago. So here is an example about the biochemical reaction or annotation basic unit or everything we annotated is based on biochemical reaction. So I just show you one specific example. The example is caspase 8 activation BID by cleavage. So this is one reactions occur in apoptosis. So from here you can see the original protein BID has I cannot see, has 195 amino residues. So this protein's N-terminal peptide can be cleaved by this so-called active caspase 8, which is a peptide. So cleavage remove the N-terminal 61 amino acid residues to activate this protein. So here, so as you can realize, so we provide a really technical detailed mechanism about this, how this protein activate another protein. Compared to another pathways, which is KEGG, so you can see the difference between all reactome and KEGG is, in KEGG, what they annotated is they just have this simple relationship, is functional, actually functional relationship. They just see Caspase-8 or 10 activated BID protein. But in our case, we actually provide, yes, molecular details.
Curators, the peer review, how do they, you said there's a focus on human reactions.
Yes.
But that you can infer that you offer like an inferred option for other animal systems, model organisms. And I'm curious to know, like, is that something that like in Reactome you can say like, I want, like when the biological reviewers are like, we think, you know, this reaction happens in a human cell.
Yeah.
Do they say like what context should they learn that from? Like what
yes.
tissue or if they're like did it in a mouse and they think it's probably the same biological reaction is gonna also happen in a human. Is that like annotated in Reactome?
Yes, this is actually the
what instructions are given to the curators as to like how confident they are?
Yeah, this is a really great question. So the majority of all the supporting evidence come from human-based experiments, but there are still some of contents actually come from no human-based experimental evidence. For example, like you said, some of the reaction come from maybe yeast or maybe mouse. So we use this so-called sequence orthologs to try to predict it. If this reaction happens in mouse, can we actually see if there's some counterpart in human, and then we predict it. So of course all this become like computation prediction. So the actual confidence is really a big question. Yeah, but that's just the state of the art. Yes. Okay. So the reason is why we want to do this is because when we see there are some human reactions at this end and then another human reaction at another end. So there's some kind of gap existing. So we can borrow all this non-human experimental evidence, try to link all this gap together, which give another evidence, see it is possible because why this happened, this also happened, so definitely there's some gap existing. So, okay. More questions? No? Okay. So come back to our annotation again. So from here you can see one reaction's output here can be used as an input or catalyst or regulators in another reactions. So therefore, by using this approach, we can connect all these reactions together to form a reaction-based network. So pathways in our annotation basically is a set of reactions connected to each other like this way. So all contents in reactome can be really represented as a single reaction network for human or other species. Then can subject to the reaction-based data analysis and visualization. So pathways in reactome actually are organized in a hierarchical way. Like here, a pathway can have sub-pathways and sub-pathways, but regardless, at the bottom level is composed of biochemical reaction only. So here is an example about the hierarchical structure of human cell cycle pathway. So you can see the hierarchical structure is quite complicated, so it can have multiple levels. So the flexibility of our data model based on reactome actually is very good. So it allows us to describe all different kind of reaction types and pathways. For example, all pathways can cover metabolism, signal transduction, or development biology, cell cycle, DNA damage and repair, and also stress response. All data model can also be actually annotated disease pathways. So currently we have annotated a lot of disease pathways. We use this so-called SBGN for systems biology graphic notation to draw a pathway diagram. So we have a set of standard compliant diagram key displayed here and draw this one. So I actually will show you more about this. Okay. So to allow users to explore our pathway content, we have this software tool called the Pathway Browser, which provides features for users to browse our pathways. So the user can view the pathway hierarchical structure here, can view the actual really biologist-friendly appealing pathway diagram for a selected pathway. For example, this is a pathway diagram for caspase activation via extrinsic apoptosis secondary. So we draw all this. So it is almost a publication quality. So everything is open. So you can actually select the site this as long as your point this actually come from Reactome. So it's fine. Okay. So this is, we call this is enhanced high-level entity view. So if you drill down, so you can actually see detailed biochemical reactions. Really try to understand the biochemical mechanism as displayed here. So this is an example. So come back to my example biochemical reaction here. So when the user selects this one specific pathway, they can see a simple text talk about this reaction or pathway, and they can check out the original literature reference we use to annotate this biochemical reaction displayed here. And also, we provide a list of proteins or small molecules or whatever we have annotated for this selected biochemical reaction. Sometimes there are some other things you can select. So you can always see all this detailed information at the bottom here. So this is just an example. So there are four proteins actually involved in this complex. Why there are only 4 proteins? Okay, that's a little bit strange. Oh, sorry, actually, 2 proteins involved in this reaction. So here you see these 2 proteins involved in this reaction actually are highlighted here in different colors. It's very difficult to see here. But there are 38 proteins total for this displayed pathway. So that is, this number means 2 and above 38, okay? So if there are some protein 3D structure available, so our users can also view all these 3D structures by linking to external PDB, which is for protein data bank. So they provide all these crystal structure. So by viewing this, basically we can get some kind of really molecular details about how these 2 proteins, they are attached to each other. For example, like how this peptide, this enzyme cut this N-terminal. So there's some molecular mechanism people can try to figure it out. Okay. So we also provide this so-called expression atlas. So we pull data from a data source called Expression Atlas, hosted at European Bioinformatics Institute. So for each individual proteins, they provide expression levels in different tissue types. Here, this column actually different tissue types is actually 2 proteins for this specific biochemical reaction. So there are a lot of information. People can really try to see, if these 2 proteins are actually expressed in some specific tissue and get some ideas if this biochemical reaction actually possibly happen. So I actually have to go back 1 step. So all pathways are actually collected from all different type of tissues or cell types. So what what you see here supposedly to happen differently in different tissue types. But what you see is all this collected. So in other words, we don't have tissue-specific pathway activity or different tissue-specific pathway available right now yet. But we are working on that. Okay, so even after almost 20 years annotation, the coverage of our database still around like 50% of total human genes. So there are still a lot of things we have not annotated, or there are a lot of things we really don't know. First, probably there's no actual experiments performed to do that, or these proteins don't have actual function. So as a website, as a web application, we also try to increase our coverage of the human genome by importing protein-protein interactions onto our pathway diagram. So we use this so-called PSICQUIC web service, which is a standard provided by the protein-protein interaction community to provide a standard format for people to do all this protein-protein network-based analysis. So we use this service and overlay protein-protein interaction onto a pathway diagram. By doing this, basically, we can increase our coverage a lot. If the original protein-protein interaction data set can cover, for example, like 95% of all human genes, so we basically can view all this. So currently, there are a lot of PSICQUIC-based service available, so we can pull all these services individually from this external service. And actually, there are almost like several billion, not billions, sorry. There are several hundred millions PPI, protein-protein interaction available goes through all these different sources. So that's a lot. This is a very valuable source. Okay, yes?
One, either tissue specific, or two, incorporate data from alternative splicing proteins, or is it this is essentially just one protein,
yes,
which encompasses all the variations of potential alternate splicing and whatever tissue it happens to come from?
Right. So we are fully aware of these issues. So one project currently actually have one overlay features we have developed is try to overlay tissue-specific gene expression data onto all these pathway diagrams, so they give you some kind of ideas of what actually happened with all these protein genes in that specific pathway, so they can get a sense of tissue-specific information. Another project is one of our PhD student, Josh. So he is not here. He tried to develop some kind of machine learning approach, tried to use this genetic pathway for all tissues, tried to really infer tissue-specific information. And regarding tissue-specific isoforms, we annotated some really small subset of proteins for their isoforms, but not that tissue-specific. But the problem is with these tissue-specific isoforms, right now there are not many information, especially experimental evidence available. So people do a lot of this large-scale omic data analysis, try to see all these isoforms. So this is more like a computational biological issue. It's not really a manipulation issue. So based on our understanding. You want to see something, Christine? No? Okay. So as I said, in our web-based tools, we also provide features to help users to do pathway enrichment analysis. This actually is the first generation pathway enrichment analysis. So it's not that much, but it is really popular and can give our users a lot of pathway-based information for their uploaded gene list. In addition to enrichment analysis, we also provide this so-called species comparison and the gene expression overlay and visualization. So called the species comparison is basically we can compare two pathways, for example, between human and yeast, see what reactions existing in human and what reactions missing in yeast. So by doing this, we can get a lot of information, especially for people use all these model organisms to do experiments. Yes?
So you said that you said that 95% of human genes are covered in some of these like protein-protein interaction databases, and then sorry. Okay, and then you said that 50% of human genes are annotated in Reactome, like in the actual pathways. Do you know if anyone has actually looked to see whether, I know a lot of these protein-protein interaction networks, they have like a magnitude, like how likely, or some numeric representation of how likely that protein-protein interaction is, and whether that number actually correlates with the genes that show up in Reactome as part of pathways. Yeah. Someone's done that.
Yes.
Okay.
Yeah, so we actually, the PSICQUIC community, they also provide some kind of score to score all these protein-protein interaction. So we can use their scores to display it here. Unfortunately, my screenshot cut this control here, so there should be a score attached to each protein interaction. So we can do kind of filtering and select certain protein interactions. Yes, yeah, more confidence. Yeah, yeah. Yes, this kind of way, yeah. Okay, so here is the feature for doing protein enrichment analysis. So let's then come back to our original 120 cancer drive genes. So here is the results for doing pathway enrichment analysis for these 127 cancer drive genes. And you can see there are a lot of pathways in Reactome enriched. So this is one specific pathway called signaling by ERB2. So here, we show the actual p-values, FDR values, and also genes come from that specific list are highlighted in this pathway diagram. So you can really see where they actually involved in this pathway diagram. By doing this, currently you can actually only manually check, but in the future we will develop some kind of mathematical modeling tool can really do all this automatic analysis, see what each individual mutations can cause a pathway activity change, which will be the topic for another. Okay. So for the power, so previously I talked about all these web-based features, but for the power users who may not necessarily want to use Reactome web-based tools, they can also download the pathways and interaction data using different data formats. So I have talked about SBGN, PSICQUIC. So we also support this BioPACS, which is the current community standard for people sharing pathway data. And also this SBML, which is the standard for system biology for people who do a lot of mathematical modeling. So we can actually export our pathways into SBML format. So things they can load into their SBML based mathematical modeling tools to do whatever they like to do. Along with this data format, we also provide a RESTful API-based web service, so people can directly call our web service and try to integrate our service into their software pipeline. So we have spent a lot of time trying to tune our web service performance, so it is quite high performant. So that actually is great. So in case you are curious how we actually do annotation, so here is a pipeline. This side is a very simple pipeline how we actually do manual curation. So basically, we recruit external biologists. We call them authors. So you can use a software tool called author tool, or they can just use Microsoft Word or PowerPoint. They annotated pathways by which actually requested by us, or they can also provide topics by themselves. In their curation or in their annotation, they should provide links to external database. For example, if they annotated some proteins, they should link to UniProt. And also, they should provide experimental evidence, which should be primary literature reference for all assertions described in their pathways. And then our internal curators will work with these external authors and use a software tool called a Curator to try to make sure the content these authors annotated consistent with our data model, and we also use software tool to ensure qualities and make sure our content is consistent. And this process may go through several steps between curators and authors before actual final release. We also ask another set of external biologists to do peer review. So as you can see, the process is just like a journal publication. So by doing this, we really try to ensure high quality for the community. So before actual release, we use a tool called slice tool to slice the release ready content into a production server, and then go through another big set of software to get our content into the final web application. Okay. So in summary, we have developed a biological pathway called Reactome. So users can access Reactome based on its website and RESTful API-based web service. And they can also download its data based on several community standard file formats. They can also perform pathway-based data analysis in our website. And the content and the software is totally open. There's no restraint to use our content and the software in either academic or industrial settings. So doing pathway-based data analysis is great, but there are still some problems. So two obvious issues here is one, in Reactome, as you remember, hopefully, all pathways are organized in a hierarchical way. So this may give us some kind of issues. For example, the actual output from our enrichment analysis actually listed is a table like here for this 127 cancer drive. But if you just focus on these four pathways, they actually presented in the same tree branch. But when you actually do this, you will not really realize, oh, what actually the relationship here. You have to go back to the actual original hierarchical tree. So we have tried to develop some kind of really user-intuitive software features address this kind of problem. But as you can see, it is still very difficult from this table list to this hierarchical. Try to really understand why these four pathways, they are all significant hit. So this is one issue. The second issue is all pathways actually, a lot of pathways inside the cell, they are cross-linked by sharing same proteins and or sharing same set of reactions. All output from one pathways can actually regulate pathway activities of another pathways. So this kind of thing cross all this cross-linked pathway relationship. So for example, these two pathways signaled by FGFI or signaled by SCF-KIT. So both of them actually are sibling annotated and signature reduction pathway, which means they are actually two different types of signaling pathways. But the question is, why these two pathways are both hit by this 127 cancer drive genes? So from here, you basically cannot really answer this type of question. So what we can do? So this is just summary about these two issues. And if you are in this field, so you definitely are aware there is this so-called network-based data analysis. So the network-based data analysis basically is try to perform systems-wide biological pathway data analysis, or in this case, we should not really call pathway data analysis, we should really call network-based data analysis. The network used by this approach cover a lot of genes, usually cover over 50% of all human genes. And currently, the network used for this type of analysis usually is protein-protein interaction network. And of course, there are a lot of gene co-expression networks available for doing that. So people use a lot of network-based algorithms. They try to find the modules, also called clusters, try to find the network patterns. So after they find all these network modules, they can do some kind of functional annotation based on enrichment analysis. They also, based on that, they can try to really find the gene signatures for biomarker discovery. And by using this, they can actually see, or they can see if there are some new disease genes can be found from this network. By trying to find, for example, find cancer drug genes or other disease modules. Yes, Jim?
So going back to what you're saying about the problem with pathways being hierarchical and
Yeah.
overlapping in nature, if you identified a gene that had a variant or mutation that you want to analyze, and it happens to be high up in the hierarchy, so as you move down, it may influence 15, 16 terminal
Yes.
reactions. Is there a best practice or a good, what's the best way to describe that pathway? Do you just go up until the point where the gene exists and then use the name for whatever that larger root of the pathway is? Or is there a best practice for that so that you don't get to the point where you're including every single reaction just because this one gene had a
So you
mutation?
mean, what you actually mean is that upstream genes in mutation can affect a lot of downstream reactions, right? Yeah. So that is what I'm talking right now. Okay. So we needed to use this network-based approach. Okay.
Okay, so you described the network.
Yes. Right. Yes. Okay. So, two issues. So the first one is hierarchical. So what we can do basically just to try to flatten all these pathways into one single network. So in your case, most likely these upstream reactions will sit on the top of this whole huge network. So then by doing all this network-based traversal, you can find what downstream reactions will be. So by doing this, you can easily find the possible impact reactions for that specific mutation. Of course, because the network itself is very, very complex, there are a lot of feedback loops. So maybe some downstream reactions may not be affected by the mutation because there are some other loops can intercept this mutation signal. Okay. Okay, the second pathway crosstalk is about the issues with pathway crosstalk. So basically, if we put all these protein or genes in the same network, so everything, all pathway, all proteins will be displayed only once in the network. So in theory, there should be no such issues at all about the pathway crosstalk. Okay. And if the crosstalk coming from interaction, so they are always displayed in the same network. Okay, so in order to support a network-based approach in the Reactome project, we have developed a method to construct a function interaction network from annotated pathways in Reactome. So basically what we did is we tried to convert a reaction into a set of functional relationship. Remember, reaction is a basic unit. If we can do this, so it is possible. We can convert the whole pathways into a relation network, into a functional interaction relationship network. And sometimes there are some complex involved in biochemical reactions. So when we have a complex, we also do this pairwise converting, basically convert all these subunits into pairwise relationship. So by these two steps, basically we can convert a pathway into a set of functional relationship. But So here actually is an example. Originally, for this specific biochemical reaction, after this converted, it comes to this simple one. So it's just like a KEGG right now. So, but KEGG, the intention in KEGG actually is separated for each individual pathways. So what I'm talking right now is to create a single network, covers all pathways annotated in the Reactome database. So the actual FI network we use actually is more than extract functional interaction from annotated Reactome pathways. Why we want to do that? Basically, we try to really increase the coverage of our network, try to really provide a comprehensive network resource for the community to do network-based data analysis and visualization. So we develop this software pipeline. In addition to Reactome, we also extract functional relationships from KEGG, Pencil pathway, NCI niche pathway, NCI barcode, and we also manually include 2 TF target intelligent data sets, try to support the gene regulatory network. And we also use a very simple machine learning approach called a naive Bayesian classify, try to collect all these pairwise relationships into all this simple machine learning approach. And we use extracted functional interaction to train and validate the naive Bayesian classify. So the final FI network actually contains 2 types of FIs. One is extracted, which should be regarded as a highly reliable FI, and another one is predicted. So we use some threshold to try to pick up only highly FIs with very high scores. Okay, so we build our FI network every year. The current version is 2018, which was built in December 2018. So here is the number. So this FI network cover about 13,500 total human genes, which is about 66%, and covers 431,000 functional interactions. And compared to our current release, which is release 69, so it covers about 11,000. So there are 24% gene coverage increase. Okay. So we have developed some approaches to try to use this Reactome FI network. So the first, I'm going to show you several examples. The first one is we can use the Reactome FI network for visualization. So for example, we can project this 341 mutated genes from this 91 TCGA glioblastoma sample and map to FI network, they are 70%, and then construct this FI network for this mapped genes. After that, we do a network clustering analysis and found these 2 modules, and then we do pathway enrichment analysis and gene ontology enrichment analysis, and found in this module, the first module actually significantly enriched genes in cytoplasma, plasma membrane, cytoplasma, plasma membrane, and cytosol. And this second module, which has 50 genes, actually significantly enriched genes in nucleus. So you can imagine, actually, you don't need to imagine. So genes in this pathway relate to signal transduction pathways, genes in this pathway relate to DNA repair or chromatin mechanism. So we did some kind of statistical analysis and found the majority, 84% glioblastoma having mutations in both of these modules, which is actually interesting. So which tells us a patient should have mutations in both of these different type of genes or different pathways in order to really have a tumor development. Okay. So we also use this FI network to try to do some kind of disease gene discovery. To do that, we basically, for each gene, so we calculate some kind of so-called gene ranking score, or for cancer gene ranking score by using a public available ranking score from Memorial Sloan Kettering, and assign scores for each genes based on their neighbors. And Then we compare the original positive data set and the random data set and find the drive genes actually having higher average ranking than Based on this one, we can develop some kind of machine learning approach and really predict if a gene is disease-related or not. So this is just a simple example. The last example I'd like to talk is we also use this ReactomeFI network for network signature discovery. So originally, remember, all FI network actually is so-called unweighted FI network. Then we can try to assign expression correlations as the weights to our weighted network, to our unweighted network, or convert this unweighted FI network as weighted network. Then we subject this weighted network to a weight-sensitive network clustering algorithm, like here it's called MCL for Markov graph clustering. So then we got a set of modules, and then use these modules as a gene signature to do some data analysis. So in this case, we use one specific module, actually is the first module. We develop our samples into two groups. The first group having high expression values in this module, then the second group. So as you can see, use 5 independent breast cancer data sets, we can get very significant p-values from survival analysis. So here are the actual genes involved in this module we used for this survival analysis. So you can see based on our functional enrichment analysis, so this module enriched genes involved in the so-called Aurora B signaling and cell cycle M phase. Aurora signaling actually related to chromosome maintenance. So this all this related to. So both of them actually related to cell proliferation. So it totally makes sense because high expression here means high cell proliferation. So there is less shorter survival time. Okay. So we compare our signature with our other published breast cancer signature, and can see our actually signature performs outperform our other published signature based on these 5 independent breast cancer data sets. So which is good. So come back to our original 127 cancer drives, cancer drives. So we did the same thing and create a FI subnetwork, and then see what actually function related. So we got 4 modules. This module is related to cell cycle, TP53, some signaling pathway, and another set of signaling pathways. So this is actually the thing we tried to answer the first question we raised about the pathway crosstalk. So I just zoom in a little bit. So this, you can see signaling by FGFR and signaling by SCF-KIT genes involved. They both actually in the same module. So no wonder why these 2 pathways actually are hit by this 127 cancer drive genes, because they actually probably most of them actually the same. They are involved in the same pathway, but used as different ways, or there are some other extra. But regardless, they are shared. Okay. So I have 3 more slides before I finish. So in summary, based on reactome pathway, we have constructed a functional intelligent network. So reaction functional intelligent network can be used for data visualization, disease gene prediction, network-based gene signature discovery, and pathway crosstalk study. And based on reactome pathway and the ReactomeFI network, we developed an app called the ReactomeFIViz for Cytoscape app. In case you don't know Cytoscape, so Cytoscape probably is the most popular standalone Java application for biological network visualization and data analysis. So we developed a Cytos app for this software. And we implement a lot of features to help our users to do network and pathway-based data analysis and visualization. So this is just a subset of this, because this is based on a paper we published 5 years ago. So there are a lot of new features available. Okay, so if you'd like, you can check our app out from this Cytoscape App Store. So, and if you want to know more, you can also check out all this. So this is my final conclusions. And see here, so this is basically also a take-home message. So I will not go step by step. Okay. So finally, this is my acknowledgement. So the project is supported by NIH for almost 20 years, and also it is supported by Canada, Ontario province, and is a joint project with a group in EBI. So it is also supported by European Molecular Biology Laboratory. So this is the whole list of a team currently working on the project. And Steve O here, he is our summer intern. He is working to integrate a machine or a natural language processing technique into our curator to try to really help our curators for some kind of automatic curation. So that's it. Questions? 4 minutes left. Yes. Thank you.
First, thank you. That was an amazing talk. I think it's a great resource. It's going to become more and more useful as informatics developers come up with innovative approaches to use this awesome data that you guys have created. But I wanted to go back briefly to the idea of predictive modeling that was touched on in a couple questions and slides, and the idea that you might use this data to predict downstream consequences of perturbations in some upstream reaction or gene, or vice versa, if you have an outcome of interest, predicting what upstream events might result in that outcome. So a concrete use case, I guess, that I'd be interested in from some of the projects I work on that deal with Fanconi anemia is the idea that acetaldehyde can be a modifier and exacerbator of that condition. And when you have increased levels of acetaldehyde, that can exacerbate Fanconi symptoms, cancer development, and blood defects. So one question that some of the projects I work on have been trying to figure out is, are there ways to predict what upstream events might reduce levels of acetaldehyde? And obviously, the data that you guys have collected with the topological networks, you might be able to ask that question. What upstream, if I want to develop a drug to target a gene or a reaction, where might I look for things that are going to ultimately decrease the levels of acetaldehyde in a cell or a specific tissue? So I'm just curious about your thoughts on the feasibility of that type of thing, given the complexity of biology and the fact that these are networks that have feedback loops and crosstalk. Is it even kind of feasible to expect that would be useful to do? And are there groups doing this or services available that are starting to think about that type of thing? Because I think it would be very valuable.
Yeah, great question. So as I said, when I first started this talk, so this is really a gentle introduction about our project. So actually we have developing some really sophisticated mathematical modeling to try to really handle all these kind of issues like you pointed out. So one mathematical modeling we have developed is called a probability graphical model, so which can integrate all this omic data together into a pathway model and try to really mimic what actually happened in the upstream, for example, some mutations, and then what downstream effect. So like in your case, or if there's some mutation happened in the upstream, so we have a mathematical model try to mimic that. Another modeling we have developed is called Boolean network-based modeling. So we are using so-called fuzzy logical model. Basically, we integrate all this possible mutation functional impact into this Boolean network, which automatically converted from our curated or manually curated pathways and really try to see. So this Boolean network model we actually have used for drug impact analysis. We just published a paper last month actually for this visualized drug impact, so in the pathway context. But currently, all this mathematical modeling is still limited to one specific pathways, so which comes to this pathway crosstalk issue. So the next step we are actually trying to work on is really build some kind of big network, saying single pathway, and try to connect several pathways together For example, one project we are working with Shannon is trying to really understand the drug resistant in ML. So we try to aggregate several pathways together to build a high-level network, and then convert them into Boolean network and see different drug or different mutation can do something like that. Yeah.
And how far do you think we are away from that dream application where I could plug in my question and then get back a
I guess probably
is
need
that 5 years, 10 years?
No,
1
need
year?
20 or 30 or 50 years. This is pretty far, far away. It's really like, yeah, the first obvious question is we don't have tissue-specific network yet, and as a whole community, we don't really have this kind of large-scale tissue-specific network or pathway available. So you can see all this simulation modeling is extremely difficult, if I say so. And also, to get a really high accuracy modeling, mathematical modeling, we need really all these parameters, which is almost not possible to get. For example, like for ordinary differential equation modeling, you need the initial concentration of these kinetical parameters. It will never be got, maybe in next 50 years. Maybe I'm too pessimistic. So we will see. Yeah, we will see. But it is the direction, and also the direction we really want to go in the future. Not in the future, next 5 years probably.
The outcome has to be, like the magnitude of like the decision of the outcome is just so, especially in like the case of like patients in cancer, it has to be so precise. Like there's not really a lot of margin for error, and the consequence of like making the wrong decision is like people's lives. Yeah.
Okay. Yes, thank you. Unfortunately, we're running out of time. So thank you very much, and hope to talk to you again next time. Yeah. Thank you.